Our first speaker is going to be Tej Sowpati, talking about ATaRVa analysis of tandem repeat variation from long-read sequencing data.
Am I audible? Yeah, okay. Good morning to all of you. I want to start by thanking Shweta and Chirag for having me here, and it's an honor to present our work to all of you. So my name is Tej. My lab at CCMB Hyderabad works in the field of computational genomics. We mostly ask questions of how genetic as well as epigenetic variation can have consequences. And we mostly focus on human genomics, but we also sometimes dive into other species. In particular, we focus on two kinds of variations. One is structural variants. So these are variants usually defined as 50 nucleotides or larger in size. And the other one is called tandem repeats, which is the topic of today's discussion. And I'm going to talk to you today about a tool that we've worked on, which can identify tandem repeat variation from long-read sequencing data. So what are tandem repeats? So shown here is an example of a trimer repeat. So as you can see, in between this sequence, there is this repeat CAG that comes again and again in tandem, right? So which is why they're called tandem repeats. So you could say that the motif size is 3 nucleotides here, it's a trimer, and the overall length of the repeat is 12 nucleotides, or 12 base pairs, because you have 4 repeating units, right? So based on this motif size alone, you could sort of classify tandem repeats into different groups. One of them, the simplest, are called homopolymers, where it's just a 1 nucleotide motif that comes again and again. So if you have a stretch of A's or C's or G's or T's, they're called homopolymers. I personally club them into the second group, but whatever. So there is another group of tandem repeats, which is what is the most famous group of tandem repeats, called the short tandem repeats. And these are usually defined as 2 to 6 nucleotide motif repetitions. And now, so far STRs were mostly focused on because that was what was easier to study with. But the field now is looking up to larger repetitions called variable number tandem repeats, which are usually defined as when the motif size is 7 nucleotides or larger. There is no upper limit. Some people put an upper limit at 100 nucleotides, but personally, I don't think we should stop at 100. If there is a 200 base pair repeat, it should still be considered a tandem repeat, right? So TRs are very interesting. So first of all, they are not few in number. Like if they are prevalent across genomes. So if you look at the human genome, you get millions of them, and they are highly polymorphic. Tandem repeats have almost an order of magnitude higher mutability or mutation rates compared to other kinds of loci in the genome. So they are highly variable. And more importantly, they show a specific kind of variation called as length polymorphism. What I mean by that? So it's like, let's say I showed this example where you have 4 repeating units. If, let's say that's what is the genotype of one individual, if you look at others, they could have a contracted version of it where there are only 3 repeating units. Or you could have expanded versions of it where you could have 5, 6, 7 or whatever. But what is more important to notice is that the difference is almost in multiples of the motif size, right? It's not just one base pair addition, 2 base pair addition. It usually happens in multiples of the motif size. So here the motif size is 3. So you see the change also happening in multiples of 3. And the reason why this happens is because of how these mutations come about. It's based on something called a strand slippage during DNA replication. But I'm not going to go into the details there. But the process of identifying how many repeating units are there at any given location is called as genotyping. And it has very interesting applications. One of the most famous is called DNA fingerprinting, where essentially what you do is you look at a certain set of 13 loci, and you ask how many repeats are present at each of these 13 loci. And turns out that particular combination of genotypes is unique to an individual. Of course, there are exceptions like monozygotic twins, etc. But by and large, if you take a specific, all the 13 genotypes, it's unique to that individual. And again, there are interesting patterns, like roughly half of these alleles come from your mother, half of them come from your father, which is why DNA fingerprinting can also be used for paternity testing, etc. So, but that's 13 loci. There is another reason why a lot of people study tandem repeats, again, mostly short tandem repeats, because they are implicated in development of neurodegenerative diseases in humans. So there is a set of disorders called as repeat expansion disorders, where if you have a tandem repeat, that expands beyond an acceptable threshold can cause disease. And one of the most common or famous examples is the Huntington's disease. So you have a CAG triplet present in the HTT gene, and usually you have about 28 to 30 copies of this. But if, let's say, it expands to something more than 35, it can lead to Huntington's disease. And even more interesting, the number or the size of expansion correlates with the severity of the disease as well as age of onset. So let's say someone who has 40 repeats is likely to develop Huntington's, much more severe form of Huntington's, and also much more early. So I think there are about 70 odd locations in the genome which are associated with disorders. But like I said, there are millions of TRs present in the human genome. So what are they doing? There have been a diverse set of studies which attributed many different roles to TRs. So for example, if you have homopolymer stretches of A's and T's, they usually contribute to nucleosome positioning. So no nucleosome actually sits at the homopolymer stretches of A. Then you can have, so these CA repeats are known to affect transcription factor binding. TRs are also shown to mediate chromatin looping, where you can, they can control long-range interactions between promoters and regulatory elements. Then there is also work showing activity of TRs as enhancer blockers, which means that if you put this TR between a promoter and an enhancer, you stop their interaction. And more recently, there is much more work going on about genome-wide analysis of TRs and their function. So one work that came out in 2019 talks about effect of TR length on proximal gene expression. So they've identified some 28,000 STRs called as eSTRs, because the length, as you can see in this graph, the length of the repeat positively correlates with how much gene expression is there of a nearby gene. And there is also evidence that it can control methylation at a locus. So this is another recent example where the repeat length is inversely correlated to the amount of methylation observed here. So now we are basically picking up more and more attributes of tandem repeats, and they still, the role of a lot of TRs is still unexplored. But again, so this is where the field is progressing. But to answer any of this, the first step to do is genotyping, which is first identifying how many repeats are present at any given locus. Now, if you're doing something targeted, like DNA fingerprinting, where you're only dealing with 13 loci, it's easy to do. You do a Sanger sequencing kind of an approach, where you just amplify those 13 loci and see what's going on. But if you want to study these at a global scale, you usually rely on whole genome data sets, right? So how does that work? So shown here is an example. Let's say these are your 3 target loci, but at a genome scale, you will have millions. Now, previously, people used to use short-read data set to understand this. So short-read data sets usually only, TRs are actually tricky to genotype because it's the same copy present at multiple places in the genome. So unless you have a neighboring flank, which is unique in nature, and unless that flank is enough, you cannot reliably map them to the genome. So which is why short-read based methods were often limited in how many TRs they could sort of give information about. And this was also dependent on the TR size, which is why most of the studies focused on short tandem repeats, because longer ones are not possible to study very well with short-read data. In contrast, if now there is advancement in sequencing technologies which offer much more longer read lengths. So this is not drawn to scale, but when I say short read, we usually talk about read lengths of 150 nucleotides, whereas in long reads you get reads of 15 kb or so, which is 100 times larger than short read data. And the advantage, as you can probably imagine, is that tandem repeat loci are now covered in full in the same read, right? You don't have to do guesswork anymore. And many times, in fact, the same read will have information of more than one tandem repeat locus, right? So this is the advantage that long reads provide. Now, of course, looking at this advantage, several tools have already been developed which can take long read data and a set of your TR coordinates and tell you, okay, at this location, this is what is the genotype. And this is a short summary of those. So the first one is called Straggler, which was developed way back in 2021. And it was focused more on identifying repeat expansions. Like I said, one of the major reasons why people study TRs is for their role in repeat expansion disorders. So this was geared more towards that. The first genome-wide approach sort of came from this particular paper, which is called Target. One disadvantage with Target is that it is licensed to be used only with PacBio data. Okay, for non-starters, there are two main long-read sequencing technologies currently. One is called PacBio sequencing, or Pacific Biosciences, and another is called Oxford Nanopore sequencing, or ONT, which is what I'll be referring to. And this was actually developed by the PacBio team, so they made it licensed to be used only if you have PacBio data. Then this is another paper which probably is the best performing until ATaRVa, I should say. So this was developed by Melissa Gimreck's group, who have actually done a lot of work on using short read data for genotyping. They've extended their short read method to sort of also deal with long reads. And there is another tool called TREAT, which uses a sort of a local assembly to infer tandem repeat genotypes. This was published in, I think, it's not visible, but November 2024 or so. So the reason why we started working on ATaRVa is because when we started using these tools, we saw that they were either inaccurate or inefficient, right? And also, like I said, one of them can only be used with PacBio data, and we were mostly at in-house dealing mostly with nanopore data, right? And our one more intuition was that, like I said, most reads have information about more than one TR locus. But all these genotypers usually go locus by locus. So once it knows that you have a set of loci you're interested in, it will fetch all the information from that locus, and it will genotype them and move on. But we asked, can't we just instead process the data read-wise? So once you pick one read from a data, instead of waiting for or reprocessing it again for the next locus, in the same time you get all the information that the read has to offer, store it, and use it where relevant, right? So that's how ATaRVa was born. So it stands for Analysis of Tandem Repeat Variation, and derived from the Sanskrit word ATaRVa. And how does it work? So initially the input is basically at least 2 files. We also need the FASTA reference file because we do a lot of sequence comparison. But the input is your alignments in BAM format. So this is basically a format which has all your reads mapped to the reference genome, and it gives you coordinate information of where each read is mapping. And then your regions of interest in a format called bed format, which basically tells you, okay, on this chromosome from here to here is my tandem repeat that I want to analyze. And it takes both of these. And once it gets this information, what ATaRVa does is it creates a map of these locations so that it knows which regions of the BAM file or which parts of the BAM file are the most relevant for the analysis, and it can only read from those locations. So the next step is read processing. Like I said, we iterate through the data read-wise, and for every read we store a bunch of information, right? So as we scan the entire read, first we store the repeat locus data. So let's say if this read harbors information for 3 different loci, we store the information for all those 3 loci in one go. Along with that, we have other metadata of the read that is stored, which is like from which chromosome coordinates it is spanning, the read is spanning, and if it has any mismatches compared to the reference, which can be a true SNP, which can be used later on for phasing, or it could be a sequencing error. At that point, we do not really differentiate both. And we also store deletions, because if you have a large deletion which spans a TR, you should be able to know that those reads cannot contribute to the TR genotype. So we also store the deletion information from there. And once we start reading more reads, if we see the same SNP again and again, we update the SNP data by how many total reads have contributed to that, what is their overall quality, and so on. So one other thing that we do is once we process all the reads, we have to do something called a realignment. The reason is, so we are taking an aligned file, but what happens is that many aligners make mistakes at repetitive region. I'm sure Chirag will disagree because Vinomap does take care of that, but a lot of people also use other aligners which do not perform very well on repetitive regions. Mainly what we see is that if you have a repeat locus and if you have an expansion of the repeat locus, the repeat is often given as an insertion sequence rather than mapping at the repeat locus. So because of this, what we do is once we take all the read information, we realign it to the reference using Smith-Waterman algorithm. So we use the SIMD implementation of that so that it's fast. So wherever we pick nearby insertions which actually match the repeat sequence, we append that to the actual repeat locus. So then we extend the repeat region based on this kind of information, and that's where the read realignment step is done. So once we do this, we have to cluster the reads into haplotypes. So ATaRVa for now is designed for diploid genomes. Human is also a diploid genome. So it expects that there are at most 2 alleles at any given location. So the idea is that now you have all this information about what all reads are present in the data and whether they can be segregated into 2 clusters which represent both these alleles, right? So how do you do that? So if you provide something called a haplotagged BAM, so haplotagged BAM is essentially where you pre-tag each read as belonging to, let's say, haplotype 1 or haplotype 2. If that kind of information is available in the BAM file, then it uses that information. But if it's not there, then what it does is it tries to use the SNP information we recorded to phase these reads. So again, depending on the average base quality, we either choose to use that SNP or we do not. So we take informative SNPs, informative here meaning heterozygous SNPs, because if you have a SNP present in all the reads, then it cannot really be used for clustering. So based on that, we then segregate them into 2 clusters wherever there is informative SNPs. Now, this is not applicable to all the locations in the genome because you will have genomes, regions of the genome where there is no heterozygous SNP present in the vicinity. So in those cases, we use a k-means clustering. So we basically take allele lengths and we ask, with a k of 2, we ask it to make 2 clusters, right? So, and we take each of these clusters to derive the consensus sequence. So we take all the reads that come from one cluster and we build a partial order alignment graph to sort of understand how they are agreeing with each other. And based on this graph, we derive the consensus TR allele sequence, and then it gets written to the output. So there is optional something called as motif decomposition that I'll explain a bit later, but this is what we do. And the output is, output is in VCF format, the standard format, and it gives a bunch of information like what is the genotype, what are their lengths, how much read support was there, what was the average quality of SNP that was used for phasing, and the decomposed sequence if you ask for it, and also the allele sequence, right? So this is the overall algorithm of ATaRVa, but how does it work, or does it work well? So to test ATaRVa, what we did is we used a sample called as 8G002. If you have worked with genomic data before, you are familiar with that sample. It's from a consortium called Genome in a Bottle. It's a widely cataloged sample. So basically, it's been studied in every way possible, and we sort of know what the sequence is supposed to be. So what we have taken is the high quality phased assembly of this sample from the Human Pangenome Consortium, and we mapped those assemblies to the GRCH38 reference genome and extracted the actual TR alleles from the assembly. So we have the paternal length and maternal length for each TR locus, and that set of genotypes is what is used as ground truth. So now the idea is that now you take data, like whole genome data set of these samples. So we have tried 3 different data sets. One is the PacBio Hi-Fi data, and then 2 versions of ONT data, nanopore data. One is called duplex data, which is slightly more accurate, actually quite more accurate than simplex data. But simplex is what routinely gets used. So we have tested it in all these 3 kinds. They are sequenced to a coverage of 30x, which is again typical. And the idea is that now you use this data to genotype a bunch of loci, compare it with the ground truth, and ask if they're matching. So, and the bunch of loci come from this particular paper, which is a catalog of 2.9 million locations, which is recently updated to be better for a benchmarking of long read data sets, right? So how does it look like? So this is one of the main results, essentially. So what is plotted on y-axis is the overall, like 100% of all the genotype loci, right? And a dark color means that the alleles match the assembly or the ground truth exactly. And the slightly lower color means that there is a mismatch of 1 base pair. So let's say if the assembly allele is 20 and we call it 19 or 21, it's off by 1 base pair, so that goes into this. And anything larger than that is shaded gray, right? And the first bar in every plot is ATaRVa, and these are on 3 different data sets, hi-fi, duplex, and simplex. And as you can see, it performs like better than both long TR and target in PacBio data, and it also does perform well on other data sets, right? So this is, and straggler and tree do not perform as well. So this is, so in all cases, it could perform better than the other tools. Now we asked, does it do it at a cost of time? So then we looked at the time, and as you can see that this is the number of loci processed per second. So the speed of ATaRVa, which is again the first bar in every case, is almost 8 times faster than the next fastest tool on PacBio dataset. And same is applicable for other datasets also. And in fact, on simplex data, we are 15 times faster than the long TR. And if you look at the memory consumption, it is still a fraction of the memory used by other tools. So with a low memory footprint and at a speed, we still achieve good accuracy in genotyping these locations. Then we went on to test other samples from HPRC data, 4 other data sets. And in almost, so here we have only done comparison with long TR and target because the other 2 were anyway not performing as well. So in all the cases, the performance of ATaRVa was significantly better than other tools. So then the other thing that we assessed is something called Mendelian concordance. The idea is that when you pick a genotype in an offspring, ideally you want, they have come from, like I said, one allele comes from your mother, one comes from the father, and you want, so every time your offspring's alleles match one of the parental alleles, you say that they're in concordance. So when they're not matching, there are 2 possible reasons. One is that it's a true de novo mutation, which also happens, but it's extremely rare. The chance of not matching is much more likely because of a genotyping error. So this is a good way of establishing whether your tool is performing well. And the way it works is, so let's say in an example locus, you have one location where the maternal alleles are 4 or 7 units or 12 and 21 base pairs. And for the father, it is 15 and 18. And in the offspring, it falls in one of these categories, where you have a perfect match of one parental allele each, or you could have a difference of 1 base pair. Again, instead of 12 and 18, we get 12 and 17. Then you have a off by 1 repeating unit. So instead of getting 12 and 15, we get 12 and 12, or a larger error, right? So again, we did this analysis on the, so it turns out HG002 is part of a trio. So you also have the father and mother information, which is 003 and 4. And we do the same analysis. We genotype all the 3 and ask how many of them are concordant. When we look at all the 2.9 million loci, the concordance is like 99.8% in all cases. The reason for this is that most loci actually are just the same as the reference alleles. So that's why we then decided to look at only those alleles which show at least one non-reference allele. And there, this is the performance. So target was not as good. ATaRVa and long TR perform better. ATaRVa is actually slightly less performing than long TR. And like I said, another applicability of this kind of a tool is in picking repeat expansions. So then we asked, is ATaRVa able to pick up repeat expansions? So we took known samples which have repeat expansions in different genes. And when we compare with the expected length, we are able to pick the repeat expansion correctly in all cases, including something like a 2.5 kb expansion in the Huntington gene, right? So even really long expansions, it's able to pick up. So I said, so there is something called motif decomposition. So the idea is that expansion, of course, affects or causes diseases because of length, but people have shown that there are other complex scenarios. Based on which motif is expanded, you can have difference in the outcome of in terms of disease. So for example, for this locus, RFC1, which causes canvas, which leads to canvas disease. So the known expansion is actually AAGGT, which is pathogenic. But you could have other variations which are only pathogenic if they are very large, or you could have a variation like this particular motif expansion, which is actually benign. So what happens is many tandem repeat genotypers just report the length of the locus. That will not allow you to do this kind of analysis. So what we have done is essentially we take, because we already report the allele sequence, we also optionally do this motif decomposition, which is essentially a motif level representation of this sequence. And for that, what we do is we have some idea of what motif it is going to be. So we first try to use a KMP algorithm to see if that motif is present. And in most cases, that is sufficient because it is matching. But wherever it is not matching, we take that and we use a approach called Rebit, which we have also worked with in a different scenario, which uses bit sets to sort of self-align these sequences and identify if there is any repeat coming up. So in the interest of time, I'll not go into the details of this, but just to showcase, based on this, we could show that in the RFC1 locus, you could see sort of benign expansions, and you could also differentiate which of them are pathogenic expansions. So like I said, this is usually benign, and this particular variation is usually pathogenic. So to summarize the talk, hopefully I've convinced you that ATaRVa is an accurate long-read genotyper, and it works well across data sets and sequencing technologies. What I've not shown you here, but I can tell you, is that it also works with short-read data, although it's not optimized for it. But of course, it does not genotype as many loci, but the accuracy is quite good. I showed that despite the accuracy, we are very efficient. I showed that it runs almost an order of magnitude faster than the next best tool. And it also had the lowest memory footprint on the tested data set. I showed that this has high clinical applicability because we get good Mendelian concordance, and it also picks pathogenic expansions accurately. And there is a lot of support, like you have native multi-threading support, which is missing in many genotypers. And we generate motif decomposition and consensus allele, and hopefully all this will be useful for people who want to use this for their studies. So I can't end my talk without thanking the people responsible for this work. So Akshay was a PhD student in the lab who has now started the project, but now moved on for his postdoc. And this work was then taken over by Abhishek. He also had a poster on this. The motif decomposition work was done by Anukrati. In fact, she also had a poster on the algorithm for that. Then Sriram helped them in doing this, some of the analysis. I also want to acknowledge the funding that we received, both from DBT and CSIR. The source code of ATaRVa is on GitHub. If you want to check it out, you can use this QR code. And we have also recently put out a preprint describing this work. And if this is the kind of work that you think you're interested in doing, please do reach out to us. And finally, I want to thank you all for patiently listening to me. If there are any questions, I'll be happy to answer.
Thank you, great talk. I have one practical question. Have you tried with some pharmacology important genes like CYP2D6 to recognize the copy number?
So we have not done a specific analysis on this, but so there are others who have tested all the pathogenic loci, and it does pick them in most cases. So there are one or two it does miss, and we are still figuring out why that is, but yeah, including CYP.
This one is there many people are actually looking for solution because it is 4.5 thousand nucleotides long, and it can have many zero or many copies, and many actually struggle to solve this problem.
Right. So
it's a really practical question.
Of course, I would say there the success would depend on the read length, essentially. So if we have something like ultra long data, we should be able to pick it up. But algorithm wise, there is nothing stopping us from being able to pick up.
Yeah, it's just a suggestion. Yeah. Thank you.
Thank you.
I'm just trying to understand the computational requirements. So how long does ATaRVa take? I know that you have shown that it's 15 times faster than the competition, but compared to the rest of the pipeline, where you would be doing, like if I were to run ATaRVa, first I will have to do sequence alignment and some kind of variant calling to get the SNPs and all that. So
no,
compared to them, how long will this take? Yeah. And what kind of machine do you need and those kind of things?
Great question. I'm glad you asked this. So everything that I've actually shown is single-threaded performance, one thing. Second is, see, alignment is something that you cannot take out from any pipeline. You will have to do the alignment part. SNP calling you don't have to do. So if it's haplotagged, we use that information. Otherwise, we identify the SNPs of our own, right? That 2.9 million loci for 30x data set using 4 CPU cores, we do in about 15 minutes. So if you use 4 CPU cores. So that's on a normal desktop computer. It doesn't need high-grade servers, it doesn't need GPUs, none of it. So that's the speed. So whereas...
So for example, how long will alignment take for the same data set to compare whether this is a bottleneck or not?
So bottleneck will still be alignment because that is a much more intensive process, but that's constant, right? I mean, all genotypers would expect you to provide an aligned file, and now that depends on what aligner you're using, what X coverage it is. Usually, I would put it and of course your machine. If you're using 32 cores, I would put it about few hours for alignment. Yeah.
Thank you.
Yeah. Be nice. Yeah, so there are two ways I can answer this. One is that, yes, you're right that at a clinical level, most of it is still short read, but that is shifting. And the idea is that more and more long read data is coming up, and you need efficient tools to study them. Like there are tools for SNP calling, SV calling, TR, whatever. So that's one part of the answer. The second thing is, we have seen this, even if you have 10x data, we are still able to accurately pick most of the loci. Of course, there is some loss of accuracy, but even at 10x, we are able to do well. We have not gone below that, but maybe it's an exercise that I can compare and tell you. Yeah.
And one more quick question, you mentioned about phase after that data. What happens if you have compared the exact genome where it is phased and unphased? How does it compare?
I am not sure I understand. Like so. Yeah. Oh, right. Haplotagged versus non. Yeah. So I think the accuracy differs by about 0.3% or so. As expected, our heterozygous loci accuracy improves by about 0.5% in haplotagged data, but otherwise overall accuracy does not change much. Yeah.
Hello, sir. Very interesting talk and very insightful. So I would like to ask, how did you check the accuracy with respect to other tools?
It's the same way. So basically the ground truth is the constant for all. So, and now you run, with the same data, you run multiple tools and you take the genotypes that they give and compare the genotypes to the ground truth.
Okay.
So, and like I said, if it matches perfectly, we put it in match. If it's off by one base pair, we put it in that, and so on.
Because I want to check whether this tool works perfectly with polyploid organisms as well.
So, okay, I can tell you that it won't, because like I said, it's geared towards diploid genomes now. We are trying to come up with a more generalized solution which works for any ploidy, but I can tell you that it won't work for polyploidy right now.
Okay,
Yeah.
but if I would like to check it with polyploids, then I may go.
Please do and let me know how it goes. Yeah,
Sure,
yeah,
sir.
yeah.
Okay,
Yeah.
thank you.
Yeah.
So, like the accuracy was not totally 100%.
Yes.
So, I just want to know where it's failing currently and what are the currently challenging regions still left.
Yeah. So there are two places usually. So in fact, I can say most of these turn out to be errors in the sequencing data itself. So if you have a motif which is very large and the motif itself has a homopolymer, and homopolymer regions are known erroneous regions for long read data, then what happens is that there is no agreement between reads. Some says that it's 5 A's, some say 6, some say 7. So whenever you have a lot of disagreement in the data itself, then we make a mistake, and that is true for all tools. The second place where it doesn't count towards accuracy is when you have a very long allele, like if you have 1,000 base pair repeat. but I've only accounted for one base pair difference. But if it's two base pair difference also, it goes into error. And in fact, we have seen that even in those long reads, 95% of them are within 10 base pairs of each other, which is less than 2% variation. Yeah, so that's why it is not 100%. Yeah.
And did you had this part wherein you are doing error correction before doing any sort of analysis?
We have not tried that, but that's an interesting strategy. I
Yes,
was talking
yeah.
to him, and I said now there is better homopolymer performance. I think I will try that out. Yeah.
Okay. Okay, and do you think it will improve the accuracy? By a
That's the
lot?
hope. Yeah.
Okay.
Yeah.
Thanks.
I think there was one last question.
You're starting with defined sets of sites. Is it possible to run for de novo?
So without telling the TR location, at present, no, but that's a good point. We will think of if we can do that. Yeah.
All right.